Hello guys, you're welcome back. So we'll move on to paper 2. Like I always say, please try to attempt the questions, then I will reveal the answer behind, because this is the answer there, that's the answer behind there, but this is the question. So please try to attempt this. So this question here is about, if it shows a food chain here. This is a food chain of yeah, it's basically a food chain anyways. It's an aquatic food chain. So the algae is fed on or is eaten by the Daphnia. Daphnia is eaten by Hydra, and Hydra is eaten by dragonfly. Please remember that this arrow does not mean what it's what, rather it means transfer of energy. Please don't forget this. You can be asked, well, what does this arrow mean? It means transfer of energy. Don't ever forget that. So what does the question here says? Which term describes Daphnia in the food chain? Apparently, I don't know what you have chosen, but what's the answer? The answer is what? Primary producer. Sorry, primary consumer. Why? Because this one is the this is the producer, primary consumer. So the first one that eats the producer is the primary consumer. The one that eats that is the secondary consumer, and the one that eats the secondary consumer is the tertiary consumer. Please remember this energy. This arrow means energy transfer. And one amazing thing is that not the whole energy is transferred, only about 10% energy is transferred. I'm going to explain that in subsequent questions, I guess. Moving on. Draw a pyramid of biomass for the food chain above. So you're meant to draw a pyramid of biomass for this food chain. So let's see what that looks like here. I hope you're attempting that. Here the answer is. So the producer is at the bottom, and this size shows it is the largest in number, sorry, in mass. So if you add all of them together, they are largest, then followed by primary consumer, secondary consumer, tertiary consumer. So this means primary, secondary, tertiary, as it were. So energy is being transferred from one level to the next, to the next, to the next. This is very important. Now, one thing you want to remember is that I wrote, if you're seeing this blue, this calorie here. What that means is an example, please. It's not like it is what it's meant to be. This means that I'm telling you that it's only 10% of energy from one trophic level that goes to the next. Yes. So if the whole energy here was, the whole biomass here is amounted to form 100 calorie, it is 10% that will go to Daphnia, which is just 100 calorie. Then all of the Daphnia put together, if the energy will be transferred to the Hydra, will be just 10 calorie. It's just 10%. Why? Because some energy is given off as heat. Number two, the organism here cannot get all of the energy here because it can't eat everything in a way, you get what I'm saying. So let's move on. So next question. Give one reason why the total biomass of Daphnia in the pond is different from the total biomass of algae. Just a bit of what we're talking about. So this is the reason why. The biomass of Daphnia is smaller because because not all the part of the algae can be digested by Daphnia. It cannot eat everything. So those one you cannot eat or digest still contain some energy in them, all right, or some mass in them. So again, Daphne will also lose some of his biomass to urea. Some of the biomass will be lost to respiration. So any of these answers is correct. I just wrote the three of them. So any of this answer is super correct. All right, moving on. Students investigated the size of population of Daphnia in the pond. So pause the video here and read through this here. So in sample 1, they got 5 and all of that. So just 5 samples were taken, and these were the numbers of Daphnia they got in the water. So calculate the mean number of Daphnia in 1 meter cube of pond water, but take into consideration that these 1 meter cube, please, is 1,000 dm cube. So you are meant to calculate that. So how do we do that? Attempt that, please. You have all of your data added together divided by the number of data. So we have 5 of them. So when you add that up, that's 70 divided by 5, we have 14. But because we've been told at the beginning that 1 meter cube is equal to 1000 dm cube, so it's going to be 1000 times 14. That gives us 14,000, which you can write as this or in standard form as this. Hope that's making sense. So next question says, genetic material is made in DNA. Which structure in the nucleus of a human contains DNA? Interesting. So you are being asked what structure contains this one. So what's your guess? So the answer to this question is chromosome. Let me quickly say this real quick. So you should know that in a cell, you have the nucleus. Nucleus contains chromosome. Chromosome contains DNA. A segment of DNA is called gene, and the gene is responsible for making protein. Please don't forget that sequence. All right, moving on. Now, figure 3 shows part of one strand of a DNA molecule. Okay, so your job here is, I guess, to identify X, Y, Z. Let me see if there's anything we have. Okay. Oh, amazingly, you're even given you're meant to choose from these. So try to choose from, let me keep that there for you. Try to choose from that. What do you think X, Y, Z is? All right, let's do this. I'm going to hide this now to show my own answer. So, well, X is deoxyribose sugar, or if you just say sugar, you're good. Each of this is deoxyribose sugar. I say it's deoxyribose sugar because it's a DNA molecule. If it was RNA molecule, I would call it a ribose sugar. And Y is nucleotide. Y is in a nucleotide. It contains phosphate, ribose and deoxyribose sugar, and a base. So those 3 things forms a nucleotide. And the last one is what? G. G is nitrogenous base. Of course, you can see that. So this, in this case here, this is guanine, thymine, cytosine, adenine. So I just try to mention each of those. So this is guanine, cytosine, thymine, adenine, guanine, thymine, adenine again, cytosine. All right, hope that made sense. So these are the answers you're meant to choose from, which you already inputted that there. So next question, a complete DNA molecule is made of 2 strands twisted around each other. What scientific term is used to describe that? Yeah, that DNA twisting around each other. There's a word used to explain that. So what is that term called? That's what you're meant to do here. All right. So let's take a look at that now. So, and that is what double helix. So it is being said that is a double helix in shape. That's what that basically means there. All right. So the next question. DNA codes for the production of proteins. A protein molecule is a long chain of amino acids. How many amino acids could be coded for by the piece of DNA shown in figure 3? So look up here. So how many pieces? So if you should, amino acid, you should ask yourself, how many bases makes one amino acid? And that is what you want to remember. Let me explain something here real quick. Don't forget that this DNA is not the one that's going to do the coding. Basically, is going to form a template for something called mRNA by a process called transcription. So that means this will form something here. There will be codes, bases form in that. So for example, now if this is mRNA, so mRNA, normally if it is DNA, it's going to be A to T, C to G. But for RNA does not have T, so it has, what it has is U. So it's going to be U here, C here, A here again, U here again, U here, then C here, A here, U here, and that will be, what's the next one? G here. All right. Now, this is mRNA. So it's asking you how many amino acids with this form. Let me say it here. 3 base makes 1 amino acid. So what you need to do here is, let me see if you can get the answer. But let me go to the answer I have here. That will be 3. How is it 3? So let's count from that picture. So this is 1. This is 2. This is 1, 2, 3, and this is 3. Oh, that made sense. So it's 3, 3, 3, 3, 3. So this 3 is 1 amino acid, amino acid, amino acid. So that's how we got the 3 there. Hope that made sense. Moving on. Label. Oh, sorry, I just went back here. Scientists have now studied the whole human genome to give to benefit of understanding the human genome. This is testing your knowledge about HGP, which is the Human Genome Project. All right. So let's take a look at what is the benefit. Think, please. Well, it helps us to diagnosis of hereditary disease is now done early. So if someone has a disease that may likely come up later in their life, you can easily tell early enough, which is something good. So you can prevent that. All right. It has helped to understand the ancestral tracing of humans as humans migrate. So even though someone, for example, I am in the UK, but let's say I'm fair, and you want to know where I came. Probably I don't. Of course, I'm obviously black, but let's assume I'm quite fair, and you want to know my origin. By taking my DNA, when you compare it with the database of that Human Genome Project, you will know I'm a black person. Yeah, so it helps us, even though we move here and there. Even the whites, some people you can trace where they came from. Even the blacks, where they came from. All right, well, that makes sense. Moving on. Okay. Next question. Phototropism is a growth. 1 second. It's a growth in response by part of a plant to light. Name one other tropism. Give the stimulus the part of the plant. Give the stimulus the plant responds to in the tropism you have named. So you're meant to mention the tropism you're mentioning, because phototropism is a type, so there's other type of tropism. And you also mention the type of responses, the stimulus it responds to. So what's your answer? This is mine. Okay, my answer is, I gave 2 answers here. So you're meant to just give 1 answer. This answer is enough to give you 2 marks. So because I mentioned geotropism or gravitotropism, which is response to gravity, all right? Then I have hydrotropism, which is response to water. So basically we have phototropism, geotropism, hydrotropism. Then we have what we call thigmotropism. Thigmotropism, or otherwise called haptotropism, which is response to mechanical obstruction. When you see plants, like you see a plant like this, so you see this is an obstacle, then a plant wraps around that. That's thigmo or haptotropism, all right? But I just feel like mentioning that right here. So this one responds to gravity, this one responds to water, that one responds to mechanical obstruction. I mean, what I just erased some seconds ago. All right, I think this diagram is for the next question, I guess. We'll come back to that. Moving on. So now, okay. Plan an investigation to show the effect of light from one direction on the growth of plant seedlings. Include details of any control needed. All right. You may use some of the equipment shown in figure 4 and any other laboratory apparatus. So you meant to use this or any other thing you feel. All right. Here's this one here. And this is a 6 marker question, so you don't want to joke with so you have to probably pause video here and think. It's just testing your understanding on phototropism. So if you understand how tropism works, majorly phototropism, and let's see. So what's your answer? So this is my own answer. It's quite long answer because that's what it demands anyways. So you should pause the video here to read through, but I'm going to quickly read through here. So seedlings are kept in different pots, of course, so like this in different pots, of course. Then the height of each of the seedlings in different pots are measured as they are sorted. So as you put them in different pots, you measure that. All right, so some of the potted plants are kept in the dark cupboard. So now you have some of the plants in the dark cupboard, cardboard or card box, but with a small hole. Please, this is meant to be a hole, please. It's meant to be a hole, like what this is what I'm trying to say here. So see it here. So you keep some directly just where the light is, nothing is taken away from them. That's like the control. And you have one covered with the cupboard, with the cardboard, I mean to say. Then this is the sun, of course. Then you create a hole. So this is the hole I was trying to tell you. Of course, let me reveal here. I'm already seeing the answer. After some time, these will bend to these. So what you are seeing here is going to deflect towards the sunlight. And there's a reason for that, which I'm going to show you towards the end of the answer there. So it says that some other potted seedling are used as control as they are exposed to equal source of light. That was the one I showed you here. This is the control. This one is the control. All right. So then these control groups are on clinostat. So I didn't draw the clinostat there, which are kept rotating now. So clinostat is an instrument you use. Oh, so I should have drawn that really. I just remember now. So clinostat looks like this. It has like some sort of handle like that. So the seedlings will be planted like that. Excuse me, that's not well drawn. Just those are the seedlings. So as you are rotating it, each of the side is getting equal light every now and then. As you're rotating it like that, it's getting equal light. Now you're going to see the effect of that later on. So now both groups are exposed to equal amount. So okay, what are we saying? So see this. So these control group are on clinostat, which keep rotating to evenly distribute light, so that every part of them receives the same amount of light. You're going to understand that. Some of you probably understand where this experiment is going to. You will know why we are doing that. I will explain anyways. So now both groups are exposed to equal amount of temperature, water, and same soil type, so that there won't be any bias. Now, change of seedling in response to light on the card box, you need to look at that, which is the one bent that way. The height of the shoot of seedlings in both groups, that means those that were exposed to light on clinostat and those that were probably on just in the cardboard, are measured again. As I say, remeasure. Now, you calculate the mean height increase of each group. Now, look at the angle of the bending to be measured, seeing that, okay, this one that bends, what's the angle to which it bends? All right, it's a 6-marker question. You are meant to write as much as you can, please. And that's why you are meant to have practice to know how to write, so that you will be able to write all of these things within a short time. Now, the group in the cardboard bent due to unequal light distribution because of one hormone called auxin. I deliberately waited to write this here. Now, auxin is a growth promoting hormone. Please work with me now. This is a part you don't want to miss out. Now, auxin likes to accumulate where there is darkness, so to say. So if I call this part A and part B, apparently this side receive more light. So by default, auxin, which is a growth promoting hormone in plant, we accumulate on this. Let me use another color to show that part. Auxin we accumulate here. So auxin having accumulated on that part will mean that this part will express more growth. So look at my hand right now. So these are the two parts. This place has more auxin. So because of the more growth, so it's going to grow more, causing this place to bend. And that is the reason why it bends that way. So it is unequal distribution of light, which makes auxin to go to the dark part And darker parts will experience more growth because auxin is a growth promoting hormone, and that will cause that part to grow longer, thereby causing it to bend. And that's why it bends to this direction. If you take the sun to this side, I can tell you, and you created a hole here, and auxin goes to accumulate on this other side, it goes the other way with time. All right, that is very important. Please, you might want to rewind the video if you have to get that part. This explanation is very important. All right. Okay, let's go back to our question. Hope that made sense. Explain how tropism in the plant should help the plant to survive. So what's the advantage of phototropism to plant? Photo, light. Yeah. So light means photo. So light is absorbed by what? Chlorophyll in the leaves, which is good. Imagine, so the way it bends towards light means it's going to absorb more light, which is beautiful. So the more the light can be absorbed, the more photosynthesis, and photosynthesis helps plants to make glucose, which is in turn used for what? Respiration and growth. Can you see the sequence? I always tell you, that's why before you start any question, think. First, analyze your answers, because usually the time given to you is enough. But what used to make it not to be enough is that when while you're writing, you're canceling, you're not sure. So you have to take out at least 30 seconds, if not more, for every question to quickly analyze. Then now start writing your answers. Make sure you have an idea of the answer in your head before you start writing. That's better. Moving on. Now, this is the next question here. The human eye can focus on objects at different distances. Figure 5 shows how a clear image of a distant object is formed in a person. So this is, let's assume there's an image of someone right there. All right. So this is object. The image is formed there in the retina. Of course, it's going to be formed upside down. It's always like that. So in our brain, this is the optic nerve. Sorry, I didn't write that very well. This optic nerve. So it's only our brain that makes it to stand upright. Usually it is upside down or inverted. And of course, this is the lens. This is the cornea. This is the suspensory ligament. This is the ciliary muscle. I can go on and on and on and on. This is the retina. This is the sclera. This is the choroid. So this is sclera, retina, choroid. All right, let me just stop right there. So what's the question asking us to do? Whoops. Explain how the person's eye could adjust to form a clear image of a nearer object. That is, so now it's all about one topic called accommodation. What's accommodation? Ability of your eye lens to change its shape to adjust to distant or close objects or near object. That's what it means. So now I'm looking at this pen right in front of me here like this. Now my eyes, there's a way it's going to adjust. Then when I'm looking far ahead of me, there's a way my eye lens is going to also adjust to do that. That's accommodation. So now pause the video and think if you can remember this topic and start reading out your point. So let me show you the answer I've written. So it's a long answer, like I told you. So it's that. So to form an image of a nearer object, the following structural adjustment is necessary. Now hear this. The ciliary muscle are connected to the suspensory ligament. Check this out. So this is, oh, that's a bit blur. This is your suspensory ligament. Then this is your ciliary muscle. All right. So ciliary muscle is connected to the suspensory ligaments, directly or indirectly, yes. Then suspensory ligament is connected to what? To the lens, because this is a lens. So you can see that connection. All right. So it says here. Okay, I think I want to do something so that I can move to see it simultaneously. Let me do that, if that will work. Okay, I don't know if that is good enough. Okay, I think that will help. I think maybe that's better, I don't know. So I hope we're going to can be able to see this. So here, Substantial ligaments are in turn connected to the lens. You can see that, the lens here. Then the ciliary muscle contracts, okay? Then the substantial ligament loosen as the diameter of the ciliary muscle is quite smaller. Let me say something here. Anytime ciliary muscle is contracting, substantial ligament is relaxing. And anytime substantial ligament is contracting, ciliary muscle is relaxing. They always work. They don't get contracted at the same time. No. When this one contracts, this one is relaxing. That is it. So now, so that's what it says here. The ciliary muscle contracts. All right. Then the substantial ligament loosen as the diameter of the ciliary muscle is quite small. So now the process will cause the lens to become more rounded. Some will say more curved. If what came to your head was curved, very correct. Now this makes the lens thicker, thereby making it more convergent. So that means it's going to make it to converge the ray of light more, because that's the idea. So once it becomes more rounded, so it's going to converge the light rays more, because now you're looking at something that is closer. You need it must converge the light rays more so that you can make to see what is closer. But if it is that, it's going to diverge more a bit. So this makes the lens thicker, thereby making it converge. This causes the light rays to focus on what? The retina. That's it. That's your answer right there. So it's just what we want to mark here is, do you understand how ciliary muscle and substantial ligament work, and what will be the effect of their contraction and relaxation on the lens? That these are key words. If I will be sincere with you, these are key answers here. Excuse me, not that one. It's part of it anyways, but I mean these ones. Everything here is super correct. I've always told you, every answer here are model answer that is worthy of accepting, taking the whole mark allotted to each question. All right, hope that made sense. So let's move on. We've done this one. Explain why a long-sighted person has difficulty seeing near objects clearly. So what does that mean? Someone that's long-sighted means they can see what is far, but they can't see what is closer to them. That's a problem. So they need something to correct that. But what is the question here is, what is wrong? What makes it hard for them to be able to see that? So what's your answer? This is because the eyeball is too short, and therefore the lens cannot be thickened enough for refractions. Okay, right, like it can't become bulgy, rounded enough, all right, for refraction. This will cause the image to form behind the retina, sadly, yes, because if people that have long-sightedness, the image is formed behind the retina. But people that have short-sightedness, the image is formed in front of the retina. I say that again. People that have long-sightedness, the image is formed behind the retina, and people that have short-sightedness, the image is formed in front of the retina. But this question is about long-sightedness, all right, which is otherwise called hyperopia. Excuse me, it's called hyperopia. On some book we call it hypermetropia. Either of this, but either of that is correct. All right. Moving on. Long-sightedness can be corrected by wearing spectacles. Describe how spectacle lens can correct long-sightedness. So here, what these people have is that the ray of light is converging late, or it diverges so much that it's not converging behind the retina, which is not nice. So you want to remember the type of lens they were going to use here, and that's the lens is going to help them correct the issue. So I took the liberty of adding this picture when I was answering this question, so that I wouldn't have to take our time to start drawing the whole thing. So this is the light ray. Can you see it? The light ray normally ought to converge somewhere here, but see where they has converged behind. So the image is going to be formed here quite blurred, which is not nice. So they now need a converging lens, which will make the light ray to converge at the right place. So this is a convex lens, which is what a converging, because the guy is converging late, so we want it to converge on time. So that's why you are using a converging lens. Don't forget that part, really. So you're using, can you see it here? Corrected hypermetropia. So you're using the convex lens, which is a converging lens. Convex lens is converging lens, while concave lens is diverging lens. If you see my video on paper 1 AQA, paper 1 Edexel, I have a question on this, on short-sightedness. You may want to check that out. I think the year is 2018 or 2016 past question. Check that up. I don't know if you have seen this on YouTube or TikTok or anywhere. Check, I uploaded this in any of these places. All right. So this is what happens. The spectacle to be used is what? A convex lens, which is what? A converging lens. This refract the overly diverged rays to convert. So to converge, please, to converge inwardly. This causes a clear image to be formed where? On the retina, period. And that's it. All right. So you pause the video, rewind it if you have to get my explanations here. Next question. Okay. So the table 2 gives the classification of 4 plants. So this is, of course, can you remember this taxa? Kingdom, phylum, class, order, family, genus, species. Even though there's something called domain before kingdom anyways, but this is not, well, I just said that, but if you're in year 12 or year 13, that's why you need to know those ones I mentioned, domain. But for year 11, GCSE, it's okay. Now, okay. What information in table 2 gives the evidence for this? So it says here, species 1 and 3 are mostly closely related. So what I would do if I were you was compare each of these for 1 and 3 and see where they have it in common. Spermatophyta, monocotyledonae, police, and that, if you look at that there, I think that's, you already know your answer right now. If you compare them with others, plantain for everybody, spermatophyta for everybody, but dicotyledonae, can you see that? So I think you know your answer right now, or maybe you're thinking of how do I write the answer. Think. All right, you have it, you know it. All right, so let's do this. The number of taxa they have in common is more, which are what? They have kingdom in common, they have order in common. The kingdom to order are the same. So the kingdom, the phylum, the class, the order are all the same. You see that? Plantae, phylum, what's it called? Excuse me. I'm talking about this. Plantae and plantae, spermatophyta and spermatophyta, monocotyledonae, then the police and police. I don't know if I pronounce that very well. All right. So moving on. So, figure 6 shows the inheritance of flower color in 2 species of plant. This is questions with genetics. Please, with inheritance and all that. Please pay attention. It's always a question you have to be calm to really understand. So this is pea plant, and this is snapdragon plant. So what we're meant to do now is this. So this is information we have. It says that in plants, please pause the video here to see the answer, to read through, so I wouldn't use a lot of time saying that. So please pause the video here, read the question, understand it, then come to the answer there. So from that question, if you understand it, you will understand the fact that this is they are all, this is homozygous for what they are, if you know what I mean. So what we're given here is this. So this big R for the red, so that means this guy is going to look like this, RR. Then this will look like this, RR, because it says for white, for the red pea plants. For this one, since they are homozygous, it's going to be CR, CR. This will be CW, CW. How do I know? ALL for white. And the reason why it is like this homozygous, because it says they are homozygous. The parental generation are homozygous for the flower. So that's why it is like this. That's why it's like this. So this is pea plant, this is snapdragon. So work with me. So what is the genotype of the red pea plant in generation? So of course, if you look at this, if you cross these, I'm talking about, I want to work with this now. Big R, you cross it with small r. If you do that or use Punnett square, you get the same answer. Big R, small r, big R, small r, big R, small r, big R, small r. So all of them will look like that. That's why the answer here looks like that. Then what's the genotype of the white pea plant? So it's the same thing. If you do that here, if I do the same thing, so it's what you're doing is this CR cross with CW. Apparently, that means all of these offspring will just be CW, just like this. It's going to be in 4 places, basically, just the same crossing. Because, and yeah, that's what's going to be. So that's what we have. Now it says question here: what's the genotype of the white? Oh, excuse me. Also, what's the genotype of the white flower? Oh, okay, it's okay. So white flower, this is it. That's why, because it's white flower. White is recessive. That's why I wrote this, and it has to be like this. So I'd forgotten. That's why I was. But of course, these are what the offspring look like. Okay, this is going to be the genotype for this. Yes, that's for pink. I am very sure. Right. Rewind the video quickly if you have to. All right. So that's I was thinking that the genotype the asked here was what was going to be here. That's why I was thinking that was the answer. But when I read it through again, of which I answered that correctly when I was answering the question. So that is so this is the genotype of this guy here. Oh, that's not too rough. Moving on. So the next question says, a gardener crossed 2 pink flower snapdragon plants. Draw a Punnett square to show why only some of the next generation of plants had pink colors, and cite the phenotype of all the offspring. Interesting. So draw a Punnett square. You know what the Punnett square is, all right? Then to show why some of the next generation only had pink color, not just some. That means not all. So let's look at what you've drawn. So this is the answer here. So this is genotype of the. It says pink. You know, I told you here. This is pink. So this is pink cross with pink. All right. So this means this, okay, yeah, this is pink. Yeah, this means this is red, this is white. So because they are together and they've crossed, it has formed pink. So this is pink. So if you cross that here, this is red homozygous. This is pink. This thing that's called pink, there's a name for it. It's called incomplete dominance. What does that mean? It means that the allele for red did not completely dominated white. So it is not purely red, it's not purely white. All right. But I know you guys probably may be used to dominance, which means that, okay, the plant is just red. Like we have here. Like all this plant here basically will be red because this guy will dominate over this. But in that case here, since we have pink, it's telling it is incomplete dominance. Well, I'm not sure it's part of the question anyway. I just mentioned that. So now, when you look at this, so this is going to be red. These two, this and this, are going to be pink. And they are heterozygous, of course. So 50% will be pink, basically. Now, so let's look at this. So how many are red? Just 1. How many are pink? That would be 2, which is this guy and this guy. How many are white? Just this 1. Hope that makes sense. All right. So it says what percentage of those will expect to have pink color? 50%. or you say 2 if the question was say in form of number, as the case may be. All right. Rewind the video if you have to. Okay. Now, commercially, hundreds of pink flower snapdragon plants can be produced from one pink flowered plant. Figure 7 shows a tissue culture technique used for that. So study this picture right now. Just look at that, all right? So that we won't waste time on that. Moving on. So if you've done that, this is your answer here. So it says here, give several reasons for the following steps. Oh, I'm sorry, I'm not sure if I allow you to see these steps. But why do you have several cells collected? This is aimed at what? Producing several plants. Yeah, that's the idea. Nutrients are added to the agar jelly. This is to make protein and amino acid available, which is essential for making the plant to grow, for making chlorophyll and all of that. All right. Then why is hormone added? If you read through that question, that briefing, you see that there's hormone added there. So this is to stimulate the cells of the plant to differentiate into stems, into root and shoot. Don't forget that shoot simply means everything above the soil, which includes the stem, the leaf, anything. So shoot just means everything above the soil, as it were. Then this is the root, as it were. All right. So the plants are kept in sterile condition. This is to prevent their entry and growth of unwanted microorganisms. Then the plants are kept at 20 degrees Celsius. This temperature is what's suitable for growth. If it is too low, it will hinder the enzymes from working, which will hinder the growth. The 20 degrees Celsius is quite optimum for this kind of condition, and it will promote the nutrients. It will denature the nutrients, and everything will work optimally. All right. Okay. Next question. Explain why the method in figure 7 produces only pink flowered plants. Why? Why will these only produce pink flower plants? We should probably... Anyways, what type of reproduction is this? That's your answer. If you remember, what type of reproduction is this? Okay. So, the reason is because the plant is sexually produced, which involves mitotic division of cells. So asexually produced organism or cell are genetically what? Identical. In this case, all plants will be what? Pink, which is this genotype of the pink, CRCW. Hope that made sense.